So I am back on Careers in Discovery with Katie Binley of Ikarovec. Katie, welcome to the show.
Hi, Tom. Nice to see you.
Great to see you. So we're recording this at the end of October. Very timely, I think, because there's been some very good news for you guys recently. Tell us a little bit about Ikarovec and where you're at, and give us an overview for those who might not be familiar.
Okay, I will do. So, Ikarovec is a gene therapy biotech company, very small one,
Hmm.
and we became operational in 2020. So we've been sort of started right in the middle of the pandemic, actually, not the best
Yes.
timing. Um, and we're focused on developing gene therapies for chronic eye diseases. So not what you might think is a classic gene therapy, where you're treating inherited retinal diseases, but
Hmm.
more delaying disease progression in chronic eye diseases like age-related macular degeneration, for
Mm.
example. Um, and so we weren't a spin out of a university, so we didn't have a big back catalogue of data. So we put a really experienced team together to put all the data together. They did a great job. I mean, we've got some fantastic data and selected some lead candidates very quickly and all of that. And we only had £3 million, which is actually, sounds like a lot, but it's not a lot in biotech. It doesn't
Right.
last very long, but we made it last about 4 years. So we
Yes.
spent the last probably year to 18 months doing a fundraise in a very difficult time, or so I'm
Mm.
told, because it's the first time I've done it. But we've managed finally, and it was announced last week that we've raised 5 million. So this is an extension of our seed round. It means that
Fantastic.
we can take the lead candidate for geographic atrophy forward. So we're really excited, and we've got some really great plans, and we've got some new people on the board as well to help us.
Yeah,
So yeah, things are looking good, very rosy.
that's great. So you talked very much at the beginning about this isn't about inherited diseases, which are clearly more obvious targets for gene therapies, right? Because there's that
Yeah, yeah,
inherent genetic component to them, but more for chronic diseases. Can you say a little bit more about that?
yeah. So the founder is Peter Widdowson, and he's got a background in pharma. So he was at AstraZeneca and
Hmm.
Pfizer, so a big company, working on small molecules for lots of different indications,
Right.
actually. And I met him when I was at Oxford Biomedica. That was his first introduction to gene therapy, and we worked together on some ophthalmic programs. And I think it's sort of really his idea is sort of using gene therapy to deliver a pharmaceutical kind of solution.
Yes.
So a sort of disease modifying approach to, to treat a condition and delay it in this case, because you're not going to
Mm.
cure age-related macular degeneration. It's not caused by genetic mutation generally, by and large. It's sort of aging and all the
Right.
things associated with that. So yeah, I mean, and it's, we're not the only ones doing it now, but I think,
Hmm.
when I first started gene therapy back in the day, when I left university, it was like it's definitely all about inherited retinal diseases, but
Yes.
actually it's so much more than that now. So yeah.
Yeah, that makes sense. So it's kind of about using the delivery mechanisms to get to the key sites of action, right? And to affect them in that way.
Yeah, and I think sort of inherited retinal diseases, fortunately don't, are rarer, generally speaking, they're
Hmm.
rarer conditions, where there's so many chronic conditions that could benefit from a gene therapy, like these eye diseases, and more people are going to be affected by them because
Yeah.
people are living longer. So, we're going to have, your eyes perhaps don't last as long as the rest of your body. So, you need
No.
treatments to preserve that vision. And it's crucial. It's so cruel. My dad's having eye injections at the moment, actually,
Right.
for this. So I'm sort of, it's very close to home for me.
Yeah, and I think people are living longer, and I think that will have an impact. But also the sort of use of computers, the use of phones, all that kind of
Oh,
stuff seems to be, everybody wears glasses now, right? Even in their 20s, everyone seems to wear glasses.
yeah, I think so. Yeah, I do. And
Yeah.
now I'm doing that thing where you kind of, I'm getting sort of short-sighted as well as long-sighted. So I'm like in between. It's awful.
Yeah, yeah, yeah. I had an eye test yesterday and I was like, oh, they're getting better. And the optician went, yeah, they'll do that. And then they'll get worse again. I was like,
Yes, I
thanks.
think I'm
Okay,
at that
cool.
stage.
Um, no, interesting. And then you talked a little bit about how you've built the company over the last 4 years. And I suppose, the point that you started at probably meant some kind of fundamental thinking about how to build a company. And you've done it with a small team with a limited budget, which, gene therapy is typically very, very expensive. Um, and then, that's allowed you to generate data and to get to a point where you've raised that extension, and that lets you take it further. But I imagine that's required some very specific thinking about what experiments to run, what things to focus on, what to prioritize, all that kind of thing that a bigger budget may not have forced, but I guess probably has instilled a bit of discipline around these things.
It has, and I think it the whole team think like this.
Mm,
Actually, we all, it's a very British thing. We try and get our value for money on everything that
right.
we do, it seems. But there comes a point which we're at now where you just have to spend quite a lot
Yeah.
of money when you get to manufacturing and large animal studies. You can't avoid it. But how we started, we're based in Norwich. So we've got a lab and an office in Norwich. I'm actually based in West Oxfordshire, which is,
Hmm.
I came here from Sheffield when I joined Oxford Biomedica back in the day in 1997, I think it was. Um, and the team are excellent. We had sort of lead scientist who was on the previous company, Quithera, so he's an ophthalmology specialist.
Right.
Andy Osborne. We've got Emily Warner and Laura Avo, who are absolutely brilliant technically in the lab and also doing disease models.
Hmm.
So we were able to. What we really focused on is running as much of the functional assays and disease models in-house as we could, because you can kind of control, the experiments a bit better and rerun them if you need to,
Yes.
and change design. And it's so cost effective. So I think being in Norwich, great facilities there. We've made fantastic use of the network. I mean, the team sort of all had links with the University of East Anglia anyway, from either PhDs or degree.
Hmm.
And so we make use of the Norwich Research Park, and they've been so supportive. So I think very cost effective being there, very sort of generous in allowing us to use bits of pieces of equipment that we just couldn't afford to buy.
Right.
So we make use of all of that, plus a disease modeling unit where we can run disease relevant models. And I think that enabled us to generate an incredible amount of data. I mean, I think it's a surprise to everybody that I speak to just how much we were able to do
Right.
in 3 years, with a very small team. So
Mm.
I just applaud them all really. We're a very tight team, but very small, but it's really effective. You don't need necessarily a big team, but you have to have a really good, strong plan, and everybody has to be on board with it. It's
Yes.
You can't have people straying off and doing other things. Everybody was very much aligned with where we needed to get to and what data we needed to be able to select something and then go out and build a narrative and
Mm.
raise money and everything. So I think, yeah, I was very lucky with that. Or engineered luck, I don't know.
Yeah, yeah. But I think those are good habits to be in as the resources become less constrained, right? It does get more expensive, and um I think the temptation is sometimes to build a bit of fat into that budget, but actually the more and things go wrong and things don't go according to plan and all that kind of stuff, and you've got to take that into account. But um that sometimes leads to a lot of waste, I think, and drug development is already expensive enough without excessive waste.
Yeah, and I think sometimes you can sort of hit a problem and think, oh, I'll bring someone else in to
Right.
solve that one, or without sort of actually looking at it yourself and really understanding what the problem is,
Mm,
seeing what you can do in-house, sort of pausing a little bit and seeing what you can do in-house, and then asking the right questions about it. And I think it
yes.
just taught us to really do it in that way. So don't rush those sort of important decisions. You have to kind of really let it settle, think
Mm,
about it, and get the right person. Because otherwise you're just going to spend a lot of money just
yeah.
and get nowhere and not even understand what the problem was fully. So I
Yes.
think it was those really important, probably quite obvious things really, but we've really utilized it, I
Mm,
think. Well,
yeah. No, it's interesting, and we'll come back to that last 4 years as we talk a bit more about your career, Katie. But I wanted to go right back to the beginning to start with, and talk a little bit about why this path for you, why science, why drug development, where did this career originate from, do you think?
oh no, how far back do you want to go? No, I would say, um, I was thinking about this before this call,
Mm,
and I moved around quite a bit when I was growing up, different schools, which was quite interesting. And one in particular, when I was 16, I moved to Doncaster from Nottingham. Never heard of
Okay.
Doncaster. And I did my A levels there. We moved in the summer, and I had my GCSE results, and I wanted to do English
Yeah.
and biology and maths, I think, but I can't quite remember, but
Okay.
definitely biology. And I went to see the new teachers, didn't know them, and they basically, and I didn't like chemistry. I didn't do particularly well in it then. And the teacher said, well, they just don't go, do they? And which one do you actually want to do? And I was thinking, well, I quite like biology. I was quite good at maths.
Mm.
Well, you need to do chemistry then, do chemistry. So I came out with, a complete different set of A levels to the ones
Yes.
I thought. But what was really amazing was that the chemistry teachers that I had, and I was quite concerned that I wasn't going to enjoy this chemistry, not one of the ones I wanted to do, had two female chemistry teachers at A level. And I wish I could remember their names, but I can't. But honestly, it was just incredible. They were
Mm.
amazing. There was three girls in our chemistry A level. But these both of these teachers were fantastic, and they really showed me that I can do this. I actually ended up being really good at chemistry, by the way.
Right.
Uh, yeah, and I loved it, and it really sort of changed it. So then I was doing three sort of science subjects, and I was loving it, and I
Mm.
loved. I was actually the chemistry was my best subject.
Okay.
And so I applied to go to Sheffield University, and I did genetics and microbiology, but it dual honours, but I could have done something very different because I really
Yes.
liked reading and writing and English. That was my thing. So I think sort of, changing schools and going
Mm.
somewhere else and really sort of opening my mind to different subjects, and then the fortunate of having those female mentors really
Yeah.
at such an early age. And I always go back to that. I think they really were changed my vision completely on what women can do.
Yes.
Um, yeah, and it was great. So I think that was the start of it. And when I got to university, we started having lectures on, I think they called it bioengineering or something,
Okay.
bioengineering lectures, genetic engineering. Gene therapy wasn't a thing. There was
Right. Yes.
there was just a little bit of, you know, getting a gene from a jellyfish to, make things glow green, things like that, and obviously there was other sort of, recombinant protein production.
Mm.
And I thought, wow, that is amazing. I want to do that. But thought, well, where the hell can you do that? I mean, is that even a thing apart
Yes.
from, lecturing about it? But I did a PhD. And yeah, I applied every Thursday. I think Nature came out and New
Mm.
Scientist. So back in the day, so take a pause on a Thursday morning to review Nature and New Scientist. That's how you looked at jobs in those days.
Right. Yes.
And apply. So apply for lots of jobs. And one of them, I've still got the advert in my filing system for Oxford Biomedica. And I saw this advert and I thought, wow, it's a gene therapy company. It was
Mm.
just setting up
Yes.
on the science park, and I applied and I got an interview, and I had an interview with Sue Kingsman, who'd come and given lectures at Sheffield actually, and everybody kind of knew her, and she, big deal in sort of yeast genetics in particular, which is what I did my PhD on.
Right.
So I had an interview with her. She was very scary, I remember, and got offered this job, and I was just like, I can't believe this has happened. I
Yes.
can't believe this has happened, and I was the first postdoc they employed. So what an opportunity that was.
Mm.
And moved to Oxford, never been there before, loved it. Being the first person in a company like that. So I started off in the biochemistry building
Yeah.
because they hadn't got the offices and the lab at the time in Oxford. They were
Okay.
just finishing it off. So the science part was just sort of growing really. So met all the team there, and I'd never done tissue culture. I'd never
Mm.
done viral vector. I hadn't done any of these things. So I just remember sort of just scrambling around and trying to do a few things to show that I kind of knew what I was doing.
Right.
And then moved to the company, and we were just setting up, the lab, putting equipment in and setting up processes. And quite quickly, I was made sort of, I think it was director of biology, which is
Mm-hmm.
basically head of research, which was great. And our team was responsible for sort of developing the new products, I guess, new ideas, trying
Right.
things
Yeah.
out. And we started off, um, looking at various, quite a broad range actually of disease areas and vector platforms. And I was known as, I think I started making adenoviruses,
Okay.
because no one else wanted to. And I said, I'll do it. So I learned how to do that. It's just, I just love doing all the new stuff. I'll have a go,
Yes.
I'll do it, and we were trying to transduce macrophages. That was the idea, because macrophages burrow into tumours, so solid tumours, and they could be sort of like a way of getting in
Hmm.
transgenes and things into the pathological region of a cancer. So it was a really
Yes.
cool idea, and they were really hard to transduce though. But
Hmm.
adenoviruses quite like them, so we did some really nice data on lots of different kind of disease models and anemia models and things like that. So I worked on something called hypoxia. Hypoxia is a sort of condition that's common in a lot of diseases like cancer,
Yes.
anemia. And then interestingly, we found out from sort of going to a lecture that it's also common in a chronic eye disease called wet age-related macular degeneration, and it really
Right.
does
Yeah.
sort of drive the pathology. And so we thought, well, we should try this promoter,
Hmm.
driving a sort of therapeutic in this condition. And we worked with Robin Alley at the Moorfields Eye Hospital, and I remember meeting his team, going to London and setting up studies with him and his team. I think it was Cam, and generated some fantastic data. And that's when I first started going to this annual conference called ARVO, which is a big eye conference that I've been sort of going to ever since,
Okay.
and just loved it. And the eye is just a wonderful organ, and it's just perfect for gene therapy. It's really needed. It's got a really unmet need, small volumes, and people really benefit. They really need this because
Yes.
sight is such a thing that people fear losing.
Yes.
So, you know, sort of done a lot of things, worked on a lot of disease areas, lots of different viral vectors, but I think I'm ending up exactly where I was meant to be
Right.
with an AAV based gene therapy and eyes and chronic eye diseases. So yeah, couldn't have worked out better, really.
No, absolutely. And you sort of touched on this, but I think there's still a lot of unknowns in gene therapy, right? We've started to see some
Oh,
gene
yeah.
therapies be approved and things like that, which is great over the last few years. But there's still lots of manufacturing challenges and lots of
Oh,
unknown
yeah.
pathways and all this kind of stuff. But when you started out at Oxford Biomedica, as you said, it wasn't even really a thing. It was just starting to come into being. So I imagine during your PhD, but also in those early years, there was just so much unexplored territory and so many new things to find out that it must have been fascinating.
Yeah, and I think anyone that's probably my age now, that's always been in this field or on the periphery of it, has probably seen all of this change
Right.
in this because also Biomedica, I remember we didn't have a quality management system.
Mm-hmm.
They didn't have manufacturing. It was, no one really knew how to, well, not many people knew how to sort of get gene therapies into clinical trial, and there wasn't that mechanism. It's like,
Yes.
the regulators were getting to grips with it. The manufacturing was, how do you do, how are you going to do this? I mean, the analytics has changed enormously over the years, so we really understand what we're actually making even
Mm-hmm.
more. We didn't then. You look back and you think it was quite basic. Um, but you just, with age and being in an industry and a field for so long, you see all those changes, and it's quite a, I guess you're part of the development of it in a small way, because you've been out
Yeah.
and you've been talking to people and giving talks and presenting, and you can see the sort of ebbs and flows in the whole field. I remember when they sequenced the whole genome in 2000. That was, until then, and it took
Right.
years. Then
Yeah.
it took years, right? And it was a bit of a shock, I think, that there weren't that many genes, and now we've discovered all this junk DNA isn't junk DNA, it's
Yes.
crucial, and all, it's just incredible. And that's 24 years, it's not that long ago, really, is it?
No.
And everybody was like doing, wanting to sort of patent genes and do genomics and all of those. So there's kind of waves of, until they find their place, there was waves of new sort of technologies, and they kind of need to sort of bed in with each other. And
Mm-hmm.
how do you use it? It's all, everybody sort of jumps on it, but actually eventually it all kind of fits together, and we're sort of using things side by side. And so I think for me, sort of, the lentivirus I used to work on, an AAV, for example, there used to be sort of like a lot of competition, which is the
Okay.
best, every conference you go to. But it's not about that. What's happened is the lentiviruses obviously fantastic for CAR T therapies, cell therapies, absolutely brilliant. Of course it is. And AAV likely has its place. So they're sort of more side by side. And I think there's this more, well, it's not a competition between the two, you know,
No.
the limited cargo capacity of an AAV. Well, you know, there's ways around that.
Mm.
Lentivirus integrates. Well, that's great if you're trying to do a cell therapy that requires it. So, you know, you can see it's sort of moved into that, you know, more
Yes.
happily sitting side by side, and depending on what you're trying to do, you pick the vectors and the components and the processes that you need.
Yeah.
And it feels a little bit more like that. I think it was all you know, very, very much developing in those sort of early 2000s. It's kind of exciting, but
Yeah.
not where it
Yeah.
is now.
No, and I think, you know, you can only take so many steps at a time, right? And, you know, learning those early things, or figuring out how to do those early, as you say, those sort of rudimentary things, was critical for all the very, very complicated things we're doing now.
Yeah, I mean, it's and to actually have gone through that
Yeah.
and learn from it and see a sort of company. I mean, Oxford Biomedica, you know, it's still around now. It's really well regarded. You know, highly involved in the AstraZeneca vaccine.
Yes.
You know, it's done amazing things, and you know, I feel really proud of the experiences that I had. I was there 25 years, and I think, you
Yes.
know, I just got the opportunity to learn so much from a lot of different people, and crucially, you know, every opportunity, you've got to take it. So probably one of the crucial things for me, where the position I'm at now, is sort of leading a sort of multidisciplinary team. So how, you know, you've got all those different, you know, facets to developing a gene therapy, you know, from doing stuff in the lab right through to sort of getting your regulatory clinical pathway sorted, but actually to see how all that fits together, and the manufacturing kind of in the middle with the, you know, tox, you know, studies that you need to worked with some brilliant people, you know, clinicians and regulatory people, manufacturing, analytical, and to see how it fits together is amazing. And I love that. It's sort of like a big jigsaw,
Right.
but you need to know when you need. The secret I think I've found is you need to know when you need to get those people in. You know, you can get
Yes.
them in too early, and then it's sort of too early and a bit of a waste perhaps. But you've got to know the questions to ask and when to ask for help in that development path. And I think we're doing that well at Ikarovec, I
Yes.
think. Because you do need help. We can't do everything with a team of 4 people,
No,
obviously, at
of
this
course.
stage.
Yeah.
But you know, you can't make those decisions lightly. You've got to get the right team together. So
Yeah, absolutely. And so thinking about that time that you had there at Oxford Biomedica, and then moving on from there, you know, 25 years is a good shift. I think, you know, anyone would say that. I guess I have two questions about it. What kept you engaged over that period of time? Because I think that's unusual to be somewhere for that long. I know the company changed a lot. Um, but then also talk us through a little bit about then that step away, and why moving into smaller biotech again. Like, what, tell us about that. These transition points I find always really interesting. So it'd be great to hear about.
yeah, it is quite interesting, isn't it? I think
Mm,
I was there because I wasn't doing the same job.
right. Yeah.
Mostly, I was constantly sort of being given different challenges, and I think that's why I stayed. And I learned a lot, and I'm glad I did. You know, so sort of being um, head of research is a really great position to be in. But I think transitioning from that to sort of program director for a Parkinson's gene therapy, which went into clinical trial, you know, that's quite a big change in a different
Mm,
disease area, multidisciplinary team. It was manufacturing, analytics, clinical. We had a big innovate project as well. So you're sort of
yes.
managing that, managing expectations outside and inside. So it sort of has its own challenges. So I think without all of the opportunities to do and learn different things, and that was just me personally, but the whole company was changing. You know,
Right.
they built manufacturing and all of the associated components of that, analytics. So you were there seeing all of these things develop right from basically, you know, nothing, from scratch.
Yes.
And not many people get to see that, actually.
No.
You know, you can move from big company to big company and just be in a little silo part of a company. But I've had an opportunity that not many people have had, and
Mm,
I recognize it, and
mm,
I've seen it, and I've, you know, sat in these, I sat in these rooms as the sort of program director, sort of listening to the whole team and thinking, this is kind of amazing. I mean, that program eventually got sold to an American company,
Right.
Which was probably one of the reasons I think it's probably one of the main reasons that I decided, now's, if I don't go now, I never will. I was
Yes.
in my sort of mid-40s. I thought, I've got to go because, yeah, otherwise I'm stuck forever. I think I need to do something new. And actually, I didn't go to Ikarovec. I went to a company called PepGen,
Mm-hmm.
which was a spin out from Oxford University, based in the bioescalator in
Yes.
Headington,
Yeah.
which interestingly was about a few hundred meters away from my first flat that I bought in Oxford.
Oh, okay.
So, and I had to use the Just Park app to park because
Yes.
there was no parking around there, and I actually parked outside in the space that was connected to my old flat, which is quite weird.
Yeah.
Uh, but anyway, and then was able to walk to work. But I was there for about a year, and I was head of preclinical. And they were working on Duchenne muscular dystrophy and
Mm-hmm.
myotonic dystrophy. So using morpholinos, which are sort of more oligonucleotides to sort of
Okay.
exon skipping. So it was quite different. And I was sort of wondering, is this gene therapy? Not, it's not quite gene therapy, but you are kind of augmenting expression in cells. So it's sort of similar.
Yes.
But that IPO'd and is now doing really well in Boston. I still follow them on LinkedIn and
Mm-hmm.
see they're doing really well. And there's a huge team being built over there. But I was there and there was just about 5 or 6 of us.
Right.
And from there, Ikarovec.
Yes.
So
So tell us about that. So tell us about how you got involved in a Ikarovec and then, obviously interesting time to join a company, as you touched on, and then you've been on this journey with them for the last 4 years, which is, recently sort of got to the point of raising this extra money, accelerating the programs now, I assume. And
that's the plan. That's the plan.
yeah, yeah. But yeah,
Well,
tell us about this sort of initial period.
well, I knew Peter Widdowson and I'd
Hmm.
remained friends with him. So he was the founder and he'd done a previous company um, that went very well actually called Quithera,
Yes.
which was span out of Cambridge University. So he was working with a guy called Keith Martin, who was a glaucoma specialist, who's now actually in Australia, heading up the Melbourne Eye Institute.
Okay.
And they worked on this sort of glaucoma idea. So it was another bicistronic gene therapy actually. So really quite clever technology, really clever idea. And they Andy Osborne worked in Keith Martin's lab and was brought in as this sort of brilliant sort of scientist to work on developing the disease models and everything, testing out the therapies, and they generated some really beautiful data.
Hmm.
And I remember getting a call from him. I think I was driving home actually from Oxford Biomedica or PepGen, and I got this call in saying, "We've got this amazing data, and Astellas, want to buy the company." And I'm like, going, "Well, go for it then. That sounds amazing." you know, And he was, I think he was, I don't know, he might have been at a conference in Hawaii or something. So he's probably
Right.
in the sea somewhere, phoning. And I was just like, "That's amazing." So but what are you going to do next? And he said, "Well, I've got all these other ideas,
Yeah.
that he just was like Mr. He's got ideas coming out of his head." So yeah, so I think I'd always sort of remained in contact with him. I think I was kind of like independent counsel,
Okay.
I don't know, and keeping him sort of, grounded. Sometimes you just need someone who's not involved with it at
Yes.
all to just talk to. And I think that was me. And it worked out quite well. And then they managed to get a little bit more funding from investors, UK investors, to set up Ikarovec and an innovate
Mm.
project. So I thought, why not give this a go? This is going to be amazing. But I'm based in West Oxfordshire, and I love it here. Although I have to say, Norwich is really nice.
Mm.
Um, Being in Oxford is very helpful. You can get into London and network and see people really easily.
Yes.
So I go over there sort of on a monthly basis. But the team, I think it's worked out quite well because the team are very sort of independently driven, and I think it sort of gives them that space because we've got a very small office and lab, and I think perhaps if I was there, maybe it just wouldn't work as well.
Right.
I think
Yeah.
it actually works quite well because you're not breathing down anyone's neck. You're kind
Yes.
of, we have our, you've got Zoom and you've got Teams and you can like discuss everything. Actually, we use Skype This is a bit weird. I don't know whether anyone else does. So we use Skype to sort of call, because it's not as personal as using sort of FaceTime, and it's not as sort of formal as perhaps Teams or Zoom.
Okay.
To set up a meeting, you can just call, and importantly, you can share the screen. So it's kind of like somewhere in between, and it's the only I only Skype my team. It's just one
Interesting.
app, and it just works really well. And actually, I used it at PetGen, because the one of the consultants suggested it to me, and I thought actually it strangely works really well. I'd recommend it. And it's just
Okay.
the only thing I do. So it's sort of separate from work, so it can be slightly
Yeah.
sort of somewhere in between,
Yeah.
somewhere not too informal, but not too formal.
No, that makes sense. But like you say, the sort of distance that you have, I guess, forces people to take some responsibility, right? And
Yeah,
means
for
that you don't
sure.
have to make every decision, because you can't, because you're not there, you're not seeing things, you're not doing all of that. So from a team development, a people development point of view, actually that physical separation can be really powerful, as long as you've got the right people in, of course.
Yeah, and you've got to trust them, and I do
Yeah.
implicitly. So I think and as long as you all know the direction you're going in, I mean, what can go wrong? I mean, we're all
Mm.
grown-ups and adults, and everybody wants it to be successful. So why wouldn't you? And everyone thinks slightly differently, which is a good thing. I don't always have the solution.
No.
It's good to sort of let people, they actually know how things work in Norwich in a way that I couldn't possibly know.
Yes.
So, it's amazing to see the sort of networks that they've built up, actually. And, I'm not sure you could necessarily do that so easily and cost effectively, perhaps anywhere else. So that was lucky, but I think it was planned and it was engineered. So I think that was the engineered
Yes.
luck I was talking about. It's not by accident, really, but I think we've really used every bit of opportunity that we could.
Mm.
Um, and yeah, and I think it's worked out well.
Yeah, and this idea of engineered luck. So I'm a big believer in it, and I think sort of um, there's always, any success story, there's always an element of fortune in it. You talked about Peter Widdowson, for example. So perhaps it's lucky that Astellas got a hold of the data and wanted to buy the company and all this kind of stuff. But they had done the right things. They had generated the data. They had
Yeah,
put themselves in a position to be lucky, right? And similar to what you've said there about the team at Ikarovec, um, yes, in some ways it's lucky that you've got the right people, but you've been deliberate about getting the right people, and so you have an opportunity to. You can't. I think the difference is you can't manufacture luck, right? You can't do it repeatably and reliably, but you can create the conditions for it. Um, and I think some people do that better than others, and it tends to be those people who are seen as kind of lucky. But
yeah, it's frustrating, isn't it? I get a bit frustrated. People say you're lucky, and you're like, are you just saying that I've just been lucky and I'm not?
yeah, yeah,
This is not planned at all, and it really is. But I mean, if for example, you're not, one thing's guaranteed: if you don't go out and meet and try and meet people, you won't meet them and you won't
yes,
network. So you've got to be there to potentially, have the opportunities to meet people and maybe build a connection, and maybe they'll be interested in collaborating or investing. So, I think you've got to engineer it, put yourself in the position for things to happen. And then, they may not always happen. It may not always work out like that, but I
no,
think you've got to be, yeah, selective about what it is that you do and where you are. And yeah, and I think I try to do that a lot in in
yeah.
in where I go, and what I attend, and who I talk to. It's it's probably more planned than it probably appears.
Yeah, and networking is a great example, right? Because you never quite know who you're going to meet. Um, but as you say, you're deliberate about what you're attending, and you're putting yourself in the right places to meet the right people, and therefore it's more likely that that luck will occur, right?
Uh, yeah, for sure. I mean,
Yeah.
probably about, I think we talked about this earlier, about 18 months ago, I never really went to many things. I didn't
Right.
really attend, and we've become, you know, I realized, you know, somebody recommended, I think it was somebody on the board, you know, you need to make sure we were members of OBN, but we're now members of One Nucleus and BIA. That was kind of like a start. You need to be part of these things, all these
Yes.
events they put on. You know, the UK is really good at putting events on. You know, actually too many sometimes. You know, you need to. You could be out all the time,
You
but I
could,
think,
yeah.
but you know, you need to go to the ones that are right, and actually you kind of meet people again, and then you build on that. And you know, I've met some fantastic, great people. I mean, my network's just exploded in the last 18 months. It's you know, but both from probably LinkedIn. You know, that was a deliberate you know, campaign that we
Yes.
decided to do to raise our profile, and I've raised my own as a consequence of that. But also networking in person. You know, you have to be there. You have to, you know, and sometimes it's, you know, it's not easy going somewhere on your own, is it?
No,
You know,
no.
but I think it's also, you're more likely to meet someone, I think, if you go on your own, because you have to, and people can
Yes.
approach you. You're more approachable on your own, aren't you? I
Yes.
think.
Yeah, absolutely. I think there's safety in numbers, right? And that's not necessarily a good
No,
thing.
it really isn't. People don't want, you know, if they see you in a couple or three, then it's sort
Yeah.
of, you're less approachable. I think if you're just kind of, you know, wandering around with your cup of coffee, it's you can have like conversations, and it's most people are really, you know, they've gone there for that reason. So I think
Yes.
you know, probably late to the party on that one, but I've realized, you know, it's you know, it's a really good thing. People are really wanting to reconnect. I don't know whether it was always like this, actually. Maybe after COVID, people wanted to see face to face. Maybe that's part of it as well. I don't know, but
Yeah, I think so. I think there's more appetite for it for sure, and sort of it because I mean there are always so many events. So people, I think before COVID got a bit fatigued on them, but there's a renewed vigor about them, I think, because everyone saw what it was like when we didn't have them, and so
everybody just wants to get out.
yeah, exactly, exactly. Um, so thinking about your career, Katie, and I guess you've talked about some of the transition points. We also skipped over basically a whole 25 years of your career through which you progressed consistently through the ranks at Oxford Biomedica and worked on lots of very interesting things. But then stepping away from that, becoming a CSO, there's lots of sort of um, points of progression, points of transition. But from a career development perspective, what do you think you've learned? What would be the advice you'd give to people earlier in their biotech career?
That's a good question. Well, I think I would, um, well, I'd maintain your network, actually.
Hmm.
I mean, it is quite valuable, and it's so easy to do. And I never really probably realized the value in that. And I think the other thing to say, it's never too late to
Right.
do that either. I mean, I probably wasn't really taking care of that, but I am now. And you can learn so much from following people, and being mentored from quite a distance, actually.
Yes.
Um, I think not rushing important decisions. I think there's sort of a tendency, because it's all, everything's emails and
Mm,
fast paced now. I think there's this idea you have to sort of make big decisions really quickly. You should, you know,
right.
if you know you're going to make a big decision. But I've really noticed since I've been sort of CSO that I need to kind of walk away
Mm,
over a weekend and sow a seed and walk away. And the number of times the solution has changed, or what I've sort of, the decision that I've come to is different from the one I might have gone with straight away, is I've lost count.
yes.
You really do need to, well, I do need to give myself time for sort of decisions, and really sort of let your brain
Hmm.
kind of work it out, because it does.
Yeah.
And don't be afraid of being pushed out your comfort zone. You know,
Yes.
take every opportunity. Don't sort of stick, because that's how you learn. I mean, you're not going to learn by doing the same thing. So if you want to progress, all of those opportunities, I don't really, you know, I was quite good at that. You know, I was just sort of, first one, who wants to learn how to make adenoviruses? I'll do it. You know,
Hmm.
I've got nothing to base that on.
Yeah.
But then no one else did, really. So, you know,
Why
what could
not?
go wrong? And you know, and go and do those presentations at conferences. It can only get better,
Mm,
you know, because it's an opportunity, and it's still nerve-wracking now. You know, I'm
yes.
in my 50s and I'm still, you know, pushed out of my comfort zone on a regular basis. But somebody was saying to me recently, but that's how you know you're learning. That's how you know you're developing, because, you know, every time you do that, there's a new, you know, facet to your, you know, to what you're able to do. And it's
Yes.
true, you know, and it might be, you know, just local events, big events, whatever, but, you know, you've got to, that shows you're learning. I hadn't really thought about it like that. I just thought, am I the only one that feels like I'm being pushed out of my comfort zone on a regular basis? But I think probably, truthfully, if people were honest, you know, maybe we're all doing that if we're learning. So don't be afraid of it.
Yeah. Yeah.
Um,
I agree. I think really good advice. And I think it's easier as you get older and, you know, more experienced to stay in your lane a little bit because there are more things in your lane, right?
yeah.
Um, but yeah, it's that constant pursuit of the new thing that's
I mean,
interesting.
I'd never done a fundraise. I'd never done
Right.
any of the things, you know, being CSO thrown in, and, you know, that there's a lot to do. I mean,
Mm,
and, you know, actually, you could probably do every little bit of it if you're only doing a little bit of it. But
yes.
when you've got the whole thing, that's huge, and it can be just massively overwhelming. But, you know, you just, you know, you have to, you can do it, you know, just to have the help you need, ask for help,
Mm,
blah blah blah. I've got a really supportive board, which is great.
yes.
And just sort of, yeah, taking on the challenges. I mean, it's exciting. I wouldn't do it if it wasn't exciting. I don't know what's next though. Maybe this will grow and we'll get a Series A. That's the plan. I'm hoping.
Yeah.
But, you know, whatever happens now, I feel like it's, you know, we've been so successful. I'm very, very proud of what I've achieved with the team at Ikarovec. You know, we've got to this stage. I couldn't, you know, that's a great feeling actually in
Mm,
itself, to feel like in a really tough field, in a tough few years. Everybody said it was really tough, the last few years fundraising. So I think you do have to sort of take a bit of pleasure in the success and achievements that
yes.
you've made, and think, yeah, that's pretty good. And I do. So
It's a really important point, and I think there's always another milestone, right? And it's easy to go from one to the next without
yeah,
stopping to realize that actually
you've
you've come
done.
pretty far.
Yeah, I mean, I've got lots of mum friends in Whitney, and they just have no, you know, they're just like, what are you doing? I don't, you know, never heard of, you know, this just like, it's quite out there. Doing gene therapy, or you're just that sort of, in that, you know, in that company, I can't pronounce the name, and
Right.
doing sort of that science that we don't really understand, and, you know, but it's everyone's different. So
Yeah, absolutely. Well, Katie, it's been brilliant to speak to you as always. Thank you so much for coming on and sharing with us, and we wish you the very best of luck with it.
thank you. Hopefully we'll be able to engineer some more luck for Ikarovec.
Absolutely, absolutely.